यहाँ पे थोड़ा सा हमने कहा थोड़ा सा रुक के इसमें बोलना है. So it means resonance energy transfer fluorescent. Means when the resonance energy will get transferred, only then fluorescent phenomena will be occurred. उसमें यही था, दो प्रोटीन पास में है, जब इसने इसको अपनी फ्लोरोसेंट एनर्जी ट्रांसफर की, तब जो ये सेकंड मॉलिक्यूल है, सेकंड प्रोटीन मॉलिक्यूल है, विल फ्लोरिश. सो रेजोनेंस एनर्जी ट्रांसफर और देन फ्लोरोसेंट. ओके, सो बट नेम for it is fluorescent resonance energy transfer. Then fourth one is FCS. वो क्या था?
Fluorescence solution
Spectroscopy. In that you will see in a particular very narrow zone, confocal zone, how much a fluorescent molecule, what you call fluctuation, means how long it will going to be reside in this zone, that you measure, that you count, so and you correlate with each other. So that's why it is a fluorescent correlation spectroscopy. It is spectroscopy, means this microscopy belongs to the electromagnetic spectrum phenomena. So this is entirely different. So that's what it is named is here. It is a spectroscopy. Rest of the things was somewhere it's a microscopy. Here it's spectroscopy, because spectrum is also, you will count the how much intensity of the fluorescent is there, how much fluctuation a particular fluorescent molecule is making. So that you count, and then you will correlate it with the another, and using some calculation, using some formula, you will create XY axis graph, and accordingly you will know the concentration as well as the, what was the second one? जो उन्होंने बताया. Okay, now after that we will move on with the scanning probe microscopy. Again, here I will tell you one word, probe. जैसे कभी-कभी गवर्नमेंट कुछ कहीं कुछ कोई घोटाला हो जाता है, कुछ अनवांटेड थिंग हो जाती है, तो गवर्नमेंट कहती है we will probe, we will investigate. Probe means investigation. So means you will probe, by going there you will investigate. So same way, scanning probe microscopy. Here a probe, what it will do, it will touch the surface and then it will create an image on computer. So that's why it is known as a scanning probe microscopy. Using a probe, you will scan the surface so that you can get the information in all three-dimensional, like X, so you can X-axis, Y-axis, and then Z-axis. So that's the beauty of this one, scanning probe microscopy. Because probe will there, I told you that rover was sent by putting so many millions of rupees. So what is the, एक satellite ऊपर जाता और उसको वो पिक्चर खींच लेता? नहीं, you need to go there physically, you need to go there. So same way, online classes and virtual means real classes में आपके बीच में भी जाके हम आपको लेक्चर दे सकते हैं, पर ऑनलाइन में we have a limitation, we cannot, कंप्यूटर के बाहर नहीं निकल के हम आपको पढ़ा सकते हैं, कंप्यूटर के ही अंदर रह के पढ़ा सकते हैं. So same way, there is a, you do have the two-dimensional. Here it is three-dimensional. I can go here and there, I can move anywhere. So it's all-dimensional. So that's what here this microscopy is, because it will scan. Again, scanning, you every day you do scanning. So you click the photo and you just take the scan. Same way, scanning using some probe, and because it's going to be a very small particle, and that's why it is functionally microscopy. Okay, so scanning probe microscopy, whatever it is written here, scanning probe microscopy covers several related technology for imaging and measuring surface on a fine scale down to the level of molecule, groups and atoms. So this is very important, because it's a microscopy, it will go to the atomic level or molecular level. So this is the beauty, and that's why it is a very sophisticated instrument. So it goes into the all direction. It can truly said that development of this technology is a major achievement for its having profound effect in the area of the science. Now I told you that there will be two scanning probe microscopy, there are several names. Here we use the probe, and two popular examples which I am going to tell you about: the scanning tunneling microscope, and second is atomic force microscope. Scanning tunneling microscope and atomic force microscope, that's we are going to discuss today. I already told you whether it's a scanning tunneling microscope or whether it's a atomic force microscope. Both have same type of instrumentation or same type of configuration which I told you at that day. That there is a probe scanner which will scan the surface. Here you can see this is the probe scanner. This is the specimen, and here there is an X control line which will help in the controlling to moving the probe on the X-axis. Then there is a Y line which will help to control to move on the Y-axis, and then this will be interaction detection mechanism. Some signal will be generated or some electric signal will be generated that will be detected through the using some electronics, and then you will see the image. Okay, so this is the common pictorial, common configuration of any scanning probe microscope. Now we will move on the scanning tunneling microscope. So if you want to cross a hill, so electron, when it shows the quantum phenomenon, it's behave different. Electron is a dual nature. One is a particle nature where it acts like a particle, and another is its wave. Okay, so in quantum, because it's a wave, so it shows quantum phenomena. And quantum phenomena, let's understand about the quantum phenomena. Let's say you want to cross a big hill. So what you have to do? You have to go up, spend so much energy, and then you will get down. The other way, you create a tunnel where you can easily pass on by using your car or whatever it is, bike, whatever you want. So where the energy is less, where you have to apply less energy, in which phenomena? Going up or going down? Tunnel, and faster as well. Okay, so quantum mechanism is like that, where electron can function like a wave, and this is, if you see in a classical view, you see this green color particle is an electron. In a classical view, electron cannot cross this barrier. Okay, cannot cross because there is a wall. Up इस wall के पास तरफ नहीं जा सकते, नहीं जा, because there is a wall barrier. But in quantum mechanism, if it is behave like a wave or quantum mechanism, it can go through this, means it can cross this barrier through the wave phenomenon. So this you might be sounds little bit strange, but in quantum mechanism is this way, where in wave phenomena it can cross, उस तरफ भी जा सकती है, it can cross the barrier using the wave phenomenon. So it means what is the conclusion for this one? It means lesser energy required to cross this barrier. So this is the tunneling phenomena. This we call as a tunneling phenomena, where electron pass through the tunnel by using less amount of energy. So that's why this is called, and this microscope using the quantum mechanism, electron wave phenomena, it's using that methodology. Okay, so that's why it is called scanning tunneling microscope, where electron, it's through the tunnel, means it goes through the tunnel by using easy amount, it can pass on. Okay, so we will see. A scanning tunneling microscope is an instrument image surface at the atomic level. Okay, so it will surface the image at atomic level, means it's a very low level. It's not that you are talking about the 10 to the power micrometer or millimeter. No, it's too less. It's atomic level. STM is based on the concept of quantum tunneling. Okay, so it will. That's why it is called tunnel current. Means I told you that when you hill you can cross by going up and down. You can make a tunnel and easily you can. So electron because it has a dual nature, it can tunnel. Electron can cross this one. So this is the quantum mechanism. Sounds strange, but this is because we are going at atomic level. So that's why we need to understand it. Got this point? Is that clear? आपको थोड़ा strange लगेगा, but this is the way this microscopy is working. In STM, good resolution is considered to be 0.0 nanomolar lateral resolution and 0.01 nanomolar at the depth resolution. It's okay. Quantum mechanisms explain the behavior of a photon and electrons. Quantum tunneling means tunneling currents occur when electrons move through a barrier that they can classically should not be able to move through. Classically, it cannot cross the barrier, but using this we call as a tunneling current. In a classical term, if you don't have enough energy to move over a barrier, you die. It means if you don't have the energy, you cannot cross this barrier, but in quantum mechanics, you can cross this barrier even if you don't have the energy. Got this point? So, what is the tunnel, and then a little bit more about the tunneling current. When a sharp metallic tip is brought very close to the surface of a conductor, it should be conductor. Whatever the surface should be conductor, and electric current can be detected due to tunneling of electron through the air gap. If this is a surface, if even there is a space, electron can move on. In classical term, it should not, because there is a gap. Electron cannot, because circuit is not complete. So electron should not. But because of tunneling current, electron can jump, and then circuit will be completed. Okay? You got this point? You got this point? जैसे एक you know the example कि आप लोगों ने कभी वो 11,000 वोल्ट वाली लाइट देखी है, ठीक है, जो 11,000, उसमें आपने एक क्वेश्चन हमेशा एक बहुत पॉपुलर क्वेश्चन है कि मंकी इज जंपिंग, मंकी जो है उसको पकड़ के नाच सकता है पर हम नहीं उसको कर सकते, क्यों? आपने नहीं सुना कि अगर चिड़िया मींस बर्ड्स कैन सिट ऑन द 11,000 वोल्ट लाइन बट वी कैन नॉट. सुना है ये? हाँ, सर्किट नहीं कंप्लीट होता. कहीं ना कहीं हम इलेक्ट्रिक का काम करते हैं तो चप्पल पहनना बहुत जरूरी है. हाँ, सो दैट इट विल नॉट, ये जो हमारा जो सर्किट है वो जैसे ही जमीन से अर्थ से मिला सर्किट पूरा हो गया और तड़क से मीन्स यू विल गेट द करेक्ट. हाँ, तो जब तक आप कुछ भी लकड़ी का कुछ भी पहने हैं तब तक सर्किट नहीं पूरा हो रहा है, तो जो भी करंट आ रही है बस वहीं तक रुक जा रही है. जैसे ही सर्किट पूरा करेगी यू विल फील द इफेक्ट ऑफ इलेक्ट्रिक. हाँ, बिजली का झटके से वोल्टेज जो है पता लगेगा. सो हियर ऑल्दो टिप इज वेरी क्लोज बट इट इज नॉट कंप्लीटिंग द सर्किट. हाँ, सर्किट नहीं कंप्लीट हो रहा, बट in quantum, because of quantum mechanics, quantum phenomena, electron can tunnel and circuit can complete it, and then you will see a electric current, and that will be displayed on your computer, and you will see the image. Got this point? This is the tunneling current. Okay? Tunneling current means, let's say, if there is a switch, if I keep little bit away, even though tunneling current is happening, then I will get the shock. You got this point? If tunneling current at molecular, so this is at atomic level. Here we are at, means we are very large level. Okay? Got this point? So that's why it is a tunneling, a scanning tunneling microscope. Let's understand this one. So here, this is almost same. So here you can see this is your sample and this is different atomic level, because electron is showing wave function. So that's why it can go to the tips. So this is your scanning probe, and here this is your sample, and this will complete the circuit. Okay, so same, you can see there is a piezoelectric tube with electrode, and there is a tip, and there is a sample, and then there is a tunneling current amplifier. So it's what it do, it just amplify the current. It's nothing. So everything is almost same, and then you will get the data processing. But here the importance is that it's too close. See, the tip and sample is too close. So your current can be tunnel. Current can go there. Okay? So the circuit will be complete and you will get the image. So there is a two mode of thing where it is a constant, either you should keep the constant current. So if you keep the constant current, then what it will be do, to keep the constant current, your tip will be get up and down. If you have a rough surface, tip will be up and down, and if you keep so there is a two way, either you can keep the constant current. So in this here you can see. So here, if you keep the constant current, then what will be happen? Your tip will be up and down, so that the current can be constant. Okay, and there is another way where you keep the height. You keep constant height means your tip will be at particular height. Okay, and then you will scan. So then you will get different current according to the surface. If it is a wavy surface, you will get the different electronic signal, and accordingly you will see, you will get the image of your surface of your specimen. Okay, got this point? So here you can see piezoelectric tube with electrode, tunneling voltage, scanning tape, tunneling current amplifier, distance control, scanning unit. So these are the basically related with the basic instrument. So it is read it constant current and constant height. Okay. Now this is about the scanning tunnel microscope. Now there is another thing, that is atomic force microscope. So two I told you, so that's why I'm telling the second one, atomic force microscope. Here the probe is little bit different. How the probe is? This is one cantilever. So cantilever, it looks like, let's say in swim pool there is a, जब कोई जंप करता है स्विमिंग पूल में, तो एक वो जंप करने वाला जो तक पहले ऐसे 2 बार 3 बार किया फिर जंप कर गया, आपने कभी देखा है? So that is kind of cantilever which can move up and down, and then you attach a probe there. Here this is, you can see this is a cantilever, and then you attach the probe. So when this probe, and this is your surface. When this probe is going here, it will go up. Okay? So when this probe will go up, this cantilever will go up. Okay? So this much you understand? As the surface is wavy, if this is here and there is a probe, it will go up and then I can go there. So same way, if surface get any rough thing or any elevated thing, it will go up, and accordingly what will happen? Here there is a laser source, and which will put laser on your probe, and then there is a photo detector. So what is the uniqueness of this thing? When this goes up, laser little bit get deflected, and maybe this photo detector, the laser beam will be detected through this laser, this point. Okay, so that is the unique thing about this one. Photo detector will get changed, detecting the photo, and accordingly it will measure the electric current. So that is about the atomic force microscope. Okay, where there is a cantilever, it has probe. When this will go elevated, the laser source will change its detector. It will go to the another detector, and accordingly you will see the image, the electric current. Okay, so this is about the atomic force microscope. And van der Waals forces, electromagnetic forces, and other forces which arise due to their physical interaction between surface system cause the cantilever to deflect. And this deflection will lead to the detection of the laser beam will change to the different photo detector. Okay. So this is about scan atomic force microscopy. Okay. So these two comes under the scanning probe microscopy. In both you see the probe is there which will scan the surface of your specimen. Okay. So advantage and disadvantage about the atomic force microscopy, you can read it. Disadvantage is atomic, so advantage, here this advantage. Okay. So advantage and disadvantage, you can read it. The resolution is better than what I told you. I told you about the basic methodology, how it works. Now you can read it. Advantage and disadvantage. That scanning speed is low. If the tips comes in the coordinate in the surface, कोई hard surface मिल गया, तो क्या हो सकता है? It can distort. Because your tip may be distort. Okay. So this is the problem with this one, atomic force microscope. Is this okay? So this is ending of the second topic. Now there is a third and last lecture which is about the super resolution microscopy. Super resolution microscopy means here your resolution is super, means you. So there is three picture you can see. First picture, this is the image of the confocal microscopy. It's very zoom level. Then the second is super resolution microscopy, which is clicked through the super resolution, where you can see each particle at separate level. Okay. And then third is also the same thing, where it will improve the resolution of this one. Say we generally we uses the camera, mobile phone. So आप 11 पिक्सल, 12 पिक्सल, 20 पिक्सल, जितने कैमरे का resolution इसका कितना है? Resolution means when you enlarge the image, how it will resolve the 2 points. So that is the point. Means the camera which you are looking for better resolution, which will give better resolution, which will give better clarity when you will spread this your image. छोटे में तो अगर एक फोटो निकाली तो अच्छा लगता है, लेकिन अगर एक बहुत बड़े बनाना है तो then it will get distorted, because resolution was not good. 2 points are not resolved very sharply. So that's why we use super resolution microscopy to see. And what is why we need super resolution microscopy? Because when at cellular level there are, even in the different organelle, there are few cellular organelles which will very close. They are very small and they are very close. So to go at a very minute level, we are using the super resolution microscopy, so that we can see not only the mitochondria but inside the mitochondria. Even inside the mitochondria, we can see 2 cristae very clearly. So that's what it is. This is the uniqueness of the super resolution. We can visualize everything inside the cell very clearly. So that is we can do using the super resolution microscope. And this was discovered by Eric Bedzic, Stephen Hell, and William Morner. So they discovered the super resolution so very early, 2014. So they got Nobel Prize for discovering this super resolution microscope. Okay. So this means earlier you may not agree with me that this is a really good thing, but once you get the Nobel Prize, you might be agree with me that yes, this discovery is really important. That's why they awarded this using this Nobel. They give the Nobel Prize, because here the advantage is that you can go at nanomolar level. You can see ribosomes, 2 ribosomes inside everything. Whatever the protein is how ribosomes are making protein. All this you can click the image of that. That is the advantage with this super resolution microscope. Okay. So here in the super resolution, one of the image which is clicked through the super resolution microscopy. So here you can see this is a picture of any plasma membrane, and then there's a nucleus, and where you can see the nuclear pore. So because nucleus is inside the cell, and it will let's say whatever the DNA, if mRNA comes out, it needs to come out to the nuclear pore, and then it will sit on the ribosomes and then make protein. So definitely there should be pore, or there should be some channel by which it comes out, or the instruction can be passed from nucleus to cellular level, okay, to the outside in the cytoplasm. So here this is the picture. So we do have the this nuclear pore, and this nuclear pore can be easily seen using this super resolution microscope. It cannot be seen through the fluorescent microscopy. It cannot be seen through the confocal microscopy, which is more sharp. Confocal microscopy is also very sharp. You cannot see the clear, but you can see clear through the super resolution microscope. So this is the unique thing about the super resolution microscopy. Resolution is very, very good. So that you can go at very low dimensional level, and you can see different cell structure which you are not able to see using the fluorescent or confocal microscope. You got this point? So this is the important point for the super resolution microscope. Now, why the point that why we cannot see too close object very clearly. So then there is a phenomena which is called diffraction. Okay, so diffraction phenomena. So resolution is limit up to 2 small objects are seen as a satellite. What is basically resolution is limit to 2 small objects to see clearly or seen as a separate. So what happens when there is a light fall? So it will show the diffraction. It will show the diffraction means light will always get diffracted. It will, because it's waves, so it will get diffracted. Let's say if there is a narrow range, if let's say light diffraction, if light is passing through a narrow slit, then what it will do, it will diffract. It should not go straight. जैसे कि let's say if here there is a bulb and which has a very light opening, so if it will go through diffracted, it should go straight, but why it is getting deflected? So that is known as a diffraction. Okay, and because of this diffraction phenomena, we cannot see at a low level 2 separate objects. Because how you are looking, you are using light to see that object. If there is no light, you cannot see. So, but light has the property when it's narrow dimension, it will get diffracted. It will not only go straight, but it will diffract. So that's what it is, the diffraction phenomenon, and this was explained by Ernest Abbe, and where he told that light can be diffracted in X and Y in Z direction. Okay, and due to this diffraction phenomena, the 2 at very low level or at very low dimension, we cannot see 2 separate objects. What this point? आपको भी कभी-कभी जब किसी की पिक्चर और जूम करके देखते होंगे कि ये कहाँ गया है, जैसे किसी ने फेसबुक पे पिक्चर लगाई, फोटो लगाई, अब आप देखना चाहते हैं कि वो पीछे क्या लिखा हुआ है, देखें यानी कहाँ पे है, ये लाइफ साइंस डिपार्टमेंट में है कि बायोटेक डिपार्टमेंट, या पीछे के जो पढ़ना चाहें जब उसको जूम करें तो कभी-कभी अगर आपका कैमरा अच्छा है then you can read whatever written behind it. So you do, there is some example which I am not telling because specifically important, so otherwise I can tell you in the last one.
So in that case, what is the difference between diffraction and reflection?
diffraction and reflection. So refraction is medium got changed. There is a two different medium. Let's say one is water, one is air. So that is the refraction. Refractive index means some density of the other medium is different. So that's why it is refracted. Okay, whether it's a return back, even light goes and it will return back, that is also refracted. Okay, so it will fall on something and then it will be getting reflected. And then if it is going inside, then the density of two medium is different. That is reflection. And diffraction is light, same media, light get diffracted. Deflected means deflect. So it will get deflected. आप इस क्लास में आपको आना है, उस क्लास में चले गए तो what is deflection हो गया थोड़ा सा, deviation, part deviation, something like that. Okay. Now I told you that about the confocal microscope. So how confocal microscope works? So basically it works on the fluorescent phenomenon where you need to excite something. So don't forget, अगर हम इतने आगे आ गए तो आप जो पहले का पढ़ा था फ्लोरेसेंट में एक्साइटमेंट और इमिशन, उसको आप भूल न जाए, उसको भी याद रखें. Okay, so what we do in the fluorescent light is passed and it will excite. So electron goes on the upper energy level and then come down and emit some energy, and that's why the fluorescent is. Exactly in the confocal, same thing is happening. So there is an excitation laser which will fall on something which is going through the objective lens, and it is falling on some specimen. Okay, and then what will happen? So let's say this is your some area of your specimen where few molecules. Let's say these are the fluorophore. Okay, so when excitation will come, these fluorophore will emit the energy. Is this clear to you? Because they are fluorescent level. So when excitation will go, it will fluoresce. So confocal, there is a difference between fluorescent microscope and confocal microscope. That confocal microscope will so fluorescent emission will be on a particular area. It focus on only one area. Okay, let's say you are reading so many subjects. You may be reading the physics, you may be reading biology, you may be reading chemistry. But when you focus on only one subject, so then clarity will be good. So same way, here in confocal microscope, it will focus on particular area. However, in the fluorescent microscopy, you look a larger area. But in confocal, you will focus only on a particular area. So this is the area where you are looking. So when excitation laser will go, these all will start emitting. Okay, got this point? Now if they will start emitting some fluorescent signal. Okay, and then they are very close. So what will happen? Diffraction will be there. You will not see all the thing in very clear manner. Okay, let's say this is the example. This one. See, all the molecules are emitting fluorescent signal, but due to all the molecules are very close and you are zooming it, you cannot see clearly. This is exactly happening. जैसे कभी let's say मैंने सबसे कुछ एक क्वेश्चन पूछा, आप सबने बोलना शुरू कर दिया, तो मैं नहीं सुन पाया. So all are start saying something, so I cannot get. But if I will say a particular person, target, then I can. It's too audible, it's too clear to me. So same way, this super resolution microscopy, what it will do, it will allow capture image one at a time. एक बार आपको सुना, फिर एक बार आपको सुना, फिर एक बार आपको सुना, और एक बार आपको सुना, तो मैंने सबका व्यू जान लिया. Okay, so one spot at a time. पहले इसकी इमेज ली, फिर इसकी इमेज ली, फिर इसकी इमेज ली, फिर इसकी इमेज ली. So although it looks like very lengthy process, but it occurred in a very quick time. So what you are capturing image one at a time. Got this point? So that you can get better clarity. So this is exactly happening in the super resolution microscope, where what it do? So this is the first one is a confocal microscope, where they are also fluorescing, but see the resolution. See, you cannot see difference between this one. But when the super resolution microscope is, each can be, the closure can be separated easily. You can be counted. Even you can count the point. In the first, the upper one, you cannot count any signal or any point. Here you can count everything, you can quantitate. Okay, so what exactly happening? Why there is this miracle is happening? Because there is another laser. What it will do? It will, for a very few second time, it will bleach that. It will stop the signaling. It will take off its fluorescent signal. Okay, so what I will do, if I ask a question and then keep everyone mum that no, don't have to say anything. Just stop to saying something, and then I will ask for one person at a time. Okay, same way. Here, this laser will block the fluorescent signal or decrease the fluorescent signal of the other molecule, and then one at a time you will see only this one. By this way, and then after this one will be covered, and this will be empty, this one will be empty. So that you will get image, means capturing the image in a very few times. So that's why there is another laser which de-bleach. So it will overlay. So only the fluorescent particle one at a time, and then you will capture the image. And that's why we got better resolution of this picture. You got the point? Is it okay? Everything is same, but just we keep one extra light. So instead of I say to you, I especially say before asking the question, I will say no, whom I should point, that person will answer. So I put an extra. I can ask a question, you all can answer, but I cannot listen. But if I want to know if let's say target is specific, so then I will say okay, don't speak all, one should speak at a time. So then I will ask, and then I can clearly visible whether you are understanding or not. So same way, there is an extra laser beam is there which will bleach the de-bleach the other signal, and then it will capture image, and this happening very quickly, so that you will get better resolution. You got this point? Is it easy? So they should not be, but it's sounds easy, but actually it is a complicated. So this is see again. This is nuclear pore, where you can see this is the structure of the nucleus, and you can clearly see the nucleus pore. So you can study in a better way. These you can study the science in a better way. Okay. And this is here. This is the confocal this is the confocal image. Confocal is the, it is a lot advanced than in comparison to the fluorescent. I told you, I showed you one picture of mine which is taken at the fluorescent microscope and taken at the confocal as well. So there you saw the confocal was too good. But when you compare confocal to super resolution, stimulated emission depletion. So it is the STED is stimulated emission depletion. So whatever the emission is there, it will deplete. Say name, stimulated emission depletion. So if you excite some fluorescent molecule, it will start emitting some signal, and that will be depleted by this. So that's why it is called as a STED. STED. Is it clear? I think clear. If it is not clear, go back, read it again, and then it will give you, because your effort is definitely required. I cannot come inside your brain and then understanding, create any doubts, repair any doubts. Okay. So Let's see another thing, same way. See, in confocal, when the excitation is there, so you excite the molecule, all molecules start giving signal. You can see dots are there, all are giving fluorescent. But when you use the stimulated emission depletion, you count one at a time. So in that way, this super resolution microscopy work. Okay. Here this is the focal image. This is the STP. The depletion result reaches the fluorescent in a circle around the central point, which effectively increases the resolution. So that's all for. So we covered this super resolution microscopy. We covered the fluorescent microscopy, and I think now we can stop the class and stop the recording.